Hello, everybody. So today we will be going over. Hello, everybody. So today we'll be going over the triple sugar iron test. Now, this one, it trips a lot of people up because my PowerPoint has a forehead again. But it trips a lot of people up because we learn it so early. We learn it even before exam 2, but it's on exam 3. So I just wanted to go over this PowerPoint and jog the memory, and do it fairly quickly. Just because I know a lot of people don't have time, and I want to clarify common questions. Okay, so first we have the first part of the PowerPoint is about the oxidase test. So the oxidase test is very important on the on exam 3. So you need to know what the reagent is, what the actual chemical process is, what it shows, as in like what the organism would be. And then you also need to know what a positive result looks like, right? So the test tests for the organism's ability to produce cytochrome oxidase, which is the catalyst of the for the oxidation of a reduced cytochrome by molecular oxygen. That's a mouthful. So oxidase, so cytochrome oxidase is an enzyme for the oxidation of a reduced cytochrome by molecular oxygen. So the name of it sounds like it makes sense, right? We have cytochrome, oxidase, oxygen, right? So that's how you can kind of remember that. It's a lot of words and they get jumbled up very quickly, but if you remember it like that, it's a little bit easier. Now you'll need to know that it's only 10 to 15 seconds you read it. It's almost an immediate reaction. If you leave it longer, like a minute or a minute and a half, you can get a false positive. You'll need to know that the reagent is tetramethyl p phenylene diamine dihydrochloride. You'll need to know that it's TMPD. This is a mutagen, teratogen, and a carcinogen. So it is bad stuff. If you are watching this in preface, for a future lab, or if you are watching this because you were trying to cram for an exam, this is why we want to throw that in a biohazard bin immediately. If you touch that, it is bad news. This test specifically finds aerobic, facultatives, microaerophiles, and it differentiates between the oxidase positive Neisseria and Pseudomonas and the Enterobacteriaceae, which are oxidase negative. So they will not show that blue color. Remember that this is the oxidase positive, right? It's very, you will know between a positive and negative. But now for the meat and potatoes of this, we have the TSI slants. Now these have a lot of complex layers to figure out what is happening on a molecular level. They differentiate the bacteria's ability to ferment 3 different types of sugar, right? It's triple sugar, 3 sugars. It's fructose, sucrose, and glucose. They also test for their ability to reduce sulfur to hydrogen sulfide, that H2S. Now this is used to identify Enterobacteriaceae, or their more common name is just gram-negative bacilli capable of fermenting glucose. This is usually bacteria in the intestines of animals, and we are animals, right? So you'll need to know the ingredients to a T. You'll need to know that they are a protein source, right? Peptones, degraded proteins. There's a red pH indicator, that's the phenol red. That one gives it that color, right? The neutral color. The 3 glucoses and their concentrations, you'll need to know that. We have glucose, at the lowest. Now, glucose is a very large molecule, which means that the amount of actual physical space it takes up is going to be very similar to 1% of lactose. So we have 0.1% of glucose, 1% lactose, 1% sucrose. So just remember that glucose is a large molecule, so we'll obviously need a lower concentration. Okay. So then we also have sodium thiosulfate. Now this is the substrate for hydrogen sulfide gas production. This is what is used in the chemical reaction. I don't think you'll need to know the chemical equation, but it is good to know. I will say that. So sodium thiosulfate is what's being used at a chemical level. Ferrous sulfate is the detection. It is very similar to the phenol red pH indicator. It does not play a role in the actual chemical equation or the chemical reaction. It is simply there for detection. It gives you the black color, right? So remember those two differences. Those get mixed up very, very easily because of the sulfates at the end. So remember that we have the slant and the butt, right? It's very funny in lab. So we have the aerobic slant and the anaerobic butt, right? The slant portion is exposed to that atmosphere. And remember, we don't screw the cap on all the way. So we continually let air in. So just remember that part. It says it will become alkaline due to the oxidative decarbonization of proteins and amino acids. That's a little much. Just remember that the deep portion is anaerobic because of just because of how we stab it. Remember, the only oxygen that is ever introduced is that stab. But if the auger itself in that slant turns any color, that means that there is an anaerobic reaction going on. Okay, so in the butt, there is glucose. So if we see a pH change, we can understand that glucose has been fermented. So I'm just trying to read this to make sure I don't tell you guys anything wrong. I think that's the gist of this. This is what it looks like, right? And also when we are looking at a TSI tube, if I could draw on my handy dandy board, if we are looking at a TSI tube, we want to look at all of the colors, as much information as we can. So if we're going to try to look at all the information possible, where did my dry erase marker go? Thank God, here it is. All right. So if we want to look at our tube, right, and we see that the slant is red, right? And for the sake of, I don't have a yellow marker, that's yellow. So if we see this, we can say, okay, pH change, right? This is still alkaline. So if this is still alkaline and this is a pH change, we can understand that this can be written as Ka. This is the slant, this is the butt. So if we see that yellow pH change, this is how we would write it. Glucose is in the butt, right? Okay. So fermentation of all three sugars would mean that we can find the other two sugars in the slant, right? So if this was all yellow, let me erase this real quick. If this was all yellow, then all of a sudden there would be an entire pH change. This pH change would hold true with the entire slant. So then we would write this as AA, right? The slant is acidic, right? Slant is acidic and the butt is acidic. So in this, I'm just going to draw this to differentiate the butt and the slant. So this houses the lactose and the sucrose. So if there was so now that we know this information, what if this was red and the top was not, right? That would mean that this bacteria specifically likes to ferment lactose and sucrose. However, it does not ferment glucose. And then this writing would change to AK. When we see that there is no reaction, no change, or it becomes more alkaline, that means that it's going to either not prefer to do sugar at all, or if this becomes yellow, in this example, it's going to prefer these two sugars over glucose. Now, in real life, in the lab, it is not always so cut and dry. Sometimes you will push a bacteria to its limit. It will say, okay, I have nothing else left. I need something to use for energy, or else I won't live. And it will, even if it doesn't like to break down sugars, it will find a way to. Let's say it only likes lactose and sucrose, but it has run out of resources. It will almost resort to glucose. Now, this is a pretty good example for E. coli. So E. coli loves lactose and sucrose. I just combined the two. They love lactose and sucrose, but glucose, not so much. It's a big molecule. It doesn't like to eat it. But if it is pushed to its limit, it will. That's why sometimes in lab, you'll see a definite yellow up here, but it'll almost seep into the butt. It might be a little bit pink on the bottom, but it could look almost completely yellow because this has been exhausted, right? This bacteria has been exhausted and it wants to live. It'll find a way. Alrighty. So, another thing that we want to know is the production of gas. So normal gas production, because remember, bacteria one of their byproducts can be gas. It doesn't necessarily mean that it's H2S, but what it can look like is either when you stab, it can look like these little like bubbles around the stab. It will look irregular because remember, this is just a needle we're stabbing with. So if it looks irregular, that could be gas production. Okay. Another way we can see gas production is if the auger is actually distended. So it has raised up and there is a gap of air at the bottom. I think it has a good example on here. Let's see. Yes. So this second tube right here, that has distension. Okay, where we see the gas formation. So this is the actual chemical equations for hydrogen sulfide production. So we see the thiosulfate, right? We see the thiosulfate that we were talking about in the beginning get reduced. Why thiosulfate reductase? Remember that anything with the ASE is an enzyme, right? It gets reduced and we have sulfite and hydrogen sulfide gas. It is usually colorless, but because we added that ferrous sulfate, it forms into an insoluble black precipitate, ferric sulfide, okay? Usually you will only see this in the bottom portion. So you can see this be still yellow, but all of this is black. That is definite H2S production. There's no doubt about it in the lab. You will never see like specks of black and be like, oh, is that it? No, it's going to be completely black. One thing to understand is that if it is black, it is yellow underneath. There's no way that bacteria that produces H2S does not ferment glucose and does not produce that yellow color. Okay. Yes. So here is the entire blank. It's just a nice little list of like different combinations that you can have. Another thing that I did want to point out is that if a slant becomes a deeper red, it means that the peptones have been used. Okay, because remember that's one of our ingredients. So if a peptone had been used, it would form a deeper, darker red than a usual one. If it is completely red, then that means that only the peptones were used. So if you're breaking down what is in a slant, we have in the slant itself, we have peptones. We have peptones, lactose, sucrose, and in the bottom we have glucose. So with this information, if you see that peptones are used, you're going to see the slant be a deeper pink than what you started with. If you see the lactose is fermented, the thing is with the slant, because these two are both sugars and they both produce the same response, you can only say that if this was yellow, you could only say that lactose and or sucrose were used as an energy source, because there's no way to differentiate these if the slant turns yellow. But since only the butt only has glucose, we can say for sure that if the butt turns yellow, then we have fermentation of glucose. Okay, and if you follow that lineage of thought and combine them to see, and you see different colors, then you will see that when looking at the chart right here, that you will find that it lines up exactly, right? So I said if the slant was red, that it was using peptones, and if the butt was yellow, it was using glucose. So if we look up at the top, we have the red slant and the yellow butt. That symbol is Ka, right? Because K stands for alkaline, A is for acidity. We see that, oh look, glucose fermentation only, peptones catabolized, right? In terms of how to notate this, you're going to use, it's going to be the slant butt formula, right? So slant / butt formula. And it's spelled with 2 T's. It's the real butt. And then you'll put either plus G for gas if you saw those cracks or the distension, or the H2S if you see that black. Remember? Okay. So H2S is technically a gas, so you will not put plus gas plus H2S. It will only be, or I'm sorry, yes you would. If you saw cracks and distension, you would put that. It would be whatever the slant is, the butt's always going to be, is always going to be A, because remember, underneath this black is going to always be yellow. So I guess this is an F. So I'll say and or. There we go. Okay. But if there is H2S production, no matter what, it's going to be slant, whatever the slant color is, / A, because of that yellow butt, plus H2S. That's how it's always going to be, is that A is going to be here, H2S is going to be here. The only difference is that sometimes the slant can be different, or you can have other gas production. Okay, so look at this, make sure that you can go through it and understand the changes and understand what no change is also. No change is just referred to as NC. It's pretty simple to remember. Okay. So here, that's a good example of the AAH2S. Oh my gosh. So with that, we understand that the butt is yellow, but the slant can differ. But in this specific situation, the slant is acidic. You guys learned about microgens as well, but I won't be going over that today. The basic gist is that if you read this, you see if a microgen has a positive or negative result, and now you can say, okay, it's negative for lysine, positive for H2S. That's the basic gist, but I hope that you guys enjoyed this one, and I will be seeing you around. Bye. If I can spell to share.